I'm going to talk to you about our plans for the implementation of Signals Notebook at AstraZeneca, and more specifically in my area where we have opportunities for enhanced formulation development. I'll cover a little bit about our current landscape of ELN products that we have at AstraZeneca, and then go more into a user case specifically aligned towards formulation development and performance for in vivo studies, and some of the challenges we have today. And then how we look towards the future with the implementation of Signals Notebook and hopefully the opportunities we see there. So within R&D at AstraZeneca today, we have 4 ELN products. Several of these are very slow. They might even crash for the user. Some areas have almost no integrated workflows, so they really just use the ELN kind of because they have to, and they use it as a data dump. There's a lot of free text write-ups, attached files. These ELN products have no connectivity, so it's really hard for us to share information between areas and cross-functional project teams. And as many of these products are much older, there's no collaboration tools in these products either. So we had a business case to replace these ELN products and move towards one ELN in R&D, and I'll tell you more about that. So just starting with a little bit of an overview of R&D at AstraZeneca and also the ELNs that we currently have. It's a representation here of the overall pipeline. We have research, development, and delivery. And at the bottom, there's 4 boxes there that kind of cover the 4 different ELN products that we have now. We have chemistry, biology, and biologics. They're mostly in this research space, not exclusively for biologics. And then there's the pharmaceutical team, where I sit. We're primarily in this development area. And actually, historically, we were one organization. There's 2 boxes there. There's Pharmaceutical Sciences on the left and Pharmaceutical Technology and Development on the right. Once upon a time, we were one unit. And so the ELN we have that really just covers the user requirements for this development space. But some time ago, we were reorganized, split into 2 organizations. I'm in Pharmaceutical Sciences, and actually our interface isn't just, we're not just working in the development area anymore. We also have supporting preclinical drug projects. And as the ELN that we have in our area was pretty much defined for this development space, it means that our team members who are working in these preclinical drug project teams find that product really inflexible and not fit for their purposes. Oh, and so the decision has been made up front that the chemistry, biology, and biologics teams, they will all be moving over to Signals Notebook in the next I think it's about a 2-year process. And we certainly have some teams in the pharmaceutical sciences space that will go probably next year. And as far as this rest of the pharmaceutical sciences in this development area, we're just still working through that, what their timelines might be. So in May, June this year, we did a really large evaluation. We were 250 scientists from across the R&D, representing different functions: chemistry, biology, biologics, pharmaceutical sciences. We had good global coverage as well. I led a team that was preclinical formulation, and we were a mixture of scientists who work with small molecules, new modalities, and also liquid nanoparticle formulations. Not only formulation scientists, but also an analytical expertise and solid state scientists. So we were quite a mixed bag of people, so to say. And so we found quite early on in the pilot that as we were formulation is at the core of what we do ultimately, that there were development needs and enhancements for formulation in Signals Notebook, and especially because we need to be able to create a formulation, recognize it as an entity. We need to know if it's been made before. Is it a unique new formulation, or is it a batch of something we've made previously? We want to be able to make samples of that formulation and either characterize that with physical chemistry tests, or that formulation might also go into in vivo studies, as I'll talk about more. So those are the sort of, and also the quite diverse and complex calculations that go into making this variety of formulations that we work with also. So to kind of demonstrate a user case here, and I'll talk about formulations for preclinical in vivo tests. I'll use the design, make, test, analyze kind of process, which is mostly commonly presented with regard to the discovery and development of compounds to become candidate drugs. But I'll just apply that design, make, test, analyze thinking with regard to the development of in vivo studies and also the formulations to support those. So in vivo, if you're not working in pharma, is when we take we test compounds in an animal setting and preclinical. So if we start off in the design corner, so we have a bioscientist. They're working in their biology ELN. They will be making a study and be setting design parameters such as the model, species, the length of their study, and the dosing regimen. Then that will need to be put into a formulation request because they need to have a formulated compound to administer to the animals. They fill out a form, but how that's delivered to our scientists can be quite, it could be via email or it could be in a project meeting, for example. So they hand over that information with their timelines, and if there is any prior knowledge that could be relevant from the project or with the chemical series, for example, we are reliant on them kind of including some of that information in that formulation request because we're on a separate ELN system and we have no connectivity between us. With regard to make or develop, in our case, the formulation scientist then sets about designing or making a formulation to fit the study parameters. We use in-house design tools, and I also wanted to illustrate that even in this formulation development, we have our own almost DMTA cycle going on as well because we also need to test and develop a formulation before it's delivered. So there could be multiple batches made, different types of formulation. These might not be in the same formulation, same ELN experiment. When we record a batch, that batch gets a batch identifier, which is really just associated with the ELN reference. And this is not a registered formulation, so the only reference we have to the formulation is an ELN batch reference. So we have that reference and we document our formulation, how it's made, those sort of process steps, and also the active, non-active components and the relationship, the amounts between those in the formulation. Then, now it's back in the bioscientist's hands again. So we were working just before in our PharmDev ELN. We then provide the formulations to the bioscientist. They're back in their bio ELN. Those batches that I was just talking about that we call our formulations on the previous slide. Now in the bioscientist world, in their ELN, these mean absolutely nothing. There's no connectivity to their workflows, and there's no way for them to access or to see how we've made that formulation. So they execute their study. There's and they can produce data like this. This graph here kind of represents that we've got 4 different formulations, but they're all containing the same compound. So the fact that when these formulations are tested, the only registered entity in that sample that they're testing is the chemical compound. And when these results are uploaded to a database, there's no information about the formulation that sort of follows through with that. So this kind of graph we cannot build out of a database. This has to be something that the bioscientist provides to us in a project meeting or an email, for example. So as far as this DMTA goes, I think it works well for the bioscientist and the chemist with their compound and their in vivo study, but for us as formulation scientists, we miss out on the sort of feedback loop, and we don't get the sort of all the data back that describes not only the compound, but how it was formulated and how that performed in an in vivo study. So just coming back a little bit to how we work, I was saying in the beginning that that we've got ELN in our area is mostly in the development area, so it's very GXP with regard to processes and ways of working. That means certainly our scientists in this preclinical space who work a lot more exploratory, supporting these preclinical experiments, find this not fit for purpose, and they have developed some non-standardized ways of working. And hopefully you appreciate now the lack of identifier that describes not only the chemical, the compound, but also the formulation, means that we have a really hard time sharing, locating, and analyzing and reusing data with regard to formulations. So during the evaluation of Signals Notebook, I said earlier that we found some limitations with regard to formulation functionality. So this was sort of vocalized within the pilot and to myself, to the project team, and also to Revity. But during that time, we were glad to be informed that Revity is indeed working on improving formulation functionality. These are two images that have been kindly provided to me by Philip Skinner, demonstrating some of the latest calculation enhancements that have been incorporated. So the top table has a list of constituents of a formulation. Each constituent there has material percentage solids associated with it. And then as you know, the planned weights are there, there's also the calculated planned solids, and then what's actually been performed in the lab, there's the real percent solids also calculated. So at the individual level, but probably more relevant value is actually the total amount at the bottom. The table at the bottom is a similar type of calculations. However, in this case, we comparing formulation 1 to formulation 2. And the difference here being there's a top binder there that has been excluded from formulation 2. And you see how that impacts the calculation or the final amount of percent solids in formulation 2. So it's really great to see that there are enhancements to formulation functionalities going on. We've been invited to be a part of the customer consortium for formulation, which is really great, and we look forward to more improvements coming and also to be a part of that sort of dialogue and ongoing improvements. So just kind of rounding off again back towards the DMTA story with regard to preclinical formulations and how we hope to work with Signals Notebook. A huge advantage to us will be actually just being on the same ELN as our colleagues in the research area, the chemistry colleagues, the bioscience colleagues, ourselves, also DMPK. We might not start off with lots of integrated workflows between ourselves, but there's opportunity for us to do so in the future. In the case where the bioscientist makes a formulation request to us, then we can build a form, or it might be an ADT, then that can be maybe put in a task, for example, and passed over to our scientists in Pharmaceutical Sciences. With regard to make, our formulation scientists are really looking forward to a user-friendly ELN that has fit-for-purpose calculations and functionality. We hope to be able to make formulation registration and to be able to assign a sample number that can also be used in our global reporting databases, which would then close that loop of information that we want to be able to have reported back from the tests that we could either perform internally or also the biological tests. So with a persistent identifier and that feedback of data and information that we currently miss, but we look forward to being able to have more structured data and also reported data that we can feed into our design tools. And when it comes to test, this is where the bioscientist has the formulation in their hand. What I didn't mention previously was that in some cases we're not handing over a pre-made formulation that's ready to go. Sometimes they're actually having to prepare the formulation themselves. So in that case they have to work from a method that we provide. So in that case also we might like to build perhaps a worksheet with instructions and check boxes. They might need to fill in some weights and some measures and pH, something like that. But also importantly, enabling that final registration steps so that this particular batch made on this particular day gets a sample number, and then that's associated with their measurements after that. So that's something we're going to have to work out because we need to make that easy for them. We don't want to make their lives too much harder, but it would bring a lot of value to us. And so from there, of course, when it comes to their analyze and their upload of data with the registered formulation that describes not only the compound but also the formulation, then we'll learn a lot more about our formulation performance. So, but just generally across the business, we're all looking forward to being on the same ELN in the future. And with that, I would like to thank a bunch of people on the left there. These were people who were a part of the preclinical formulation cohort that I was leading, and also some line management and digitization department colleagues with a lot of discussions and support, and then the project team and Revity as well. There was a lot of support from Ben during the evaluation earlier this year, and thanks to Philip, Michelle and Lawrence for letting us know about the formulation enhancement plans and bringing us into the consortium. And thank you. With that, if the formulation topic interests you and you would like to talk more, please just reach out. And